Thank you for the introduction. And today I'm very happy to be invited and to take a plunge with you into the dark, deep ocean where we go from the surface down to over 1,000 meters deep where no light can reach. And what lives there? Lots of mollusks, it turns out. And I'm going to talk about diversity and adaptation of mollusks from deep sea chemosynthesis-based ecosystems. And I'm Chong Chen from Jamstec, where I'm a senior scientist, and I'm also an ambassador for the Zenhibird Museum's Zenhibird Ocean Species Alliance. So this is what actually most of the deep sea looks like. There isn't very many big things that's alive. And that's because the deep sea has an energy problem, and because the life in normal deep sea depends on photosynthesis, and light does not reach the deep sea, which means there's no primary production at the deep sea. So until the Challenger expedition in the late 1800s, some people thought there's no life in the deep sea. And now we know to be very different because we found lots of life on the Challenger expedition. But indeed, most deep sea life depends on organic particles that sink from the surface for energy in the form of marine snow, which means most of the animals there are tiny because there's a limitation of energy and food, so they can't grow very big. But does that mean the deep ocean can't actually make its own energy? As people thought so until 1977. So we traveled to the Galapagos, where it is famous for inspiring Darwin for his theory of evolution with giant tortoises and also the marine iguanas, but also hot vents. In 1977, a geology cruise that aimed to discover what they thought must be hydrothermal geysers in the deep sea floor. And serendipitously, they found animals that they never expected to. So here we see there are giant clams and there are giant tube worms that's much, much bigger than any of the deep sea life, any of the deep sea benthic invertebrates that was known before from 2,500 meters deep in Galapagos. And they live just beside where hot water is spewing out from the crust containing toxic fumes of hydrogen sulfide. So this is what they saw. They saw fields and fields of giant clams. So I have one of these clams here, so I'll show you how big it is, and it's pretty big. And also mountains and mountains of mussels living with these tube worms, and lots of animals that are living on both of on all of these animals. And these vents are spectacular because they can form 3D structures known as chimneys, where the minerals contained in the hot fluid rapidly precipitates as it contacts the cold seawater around it, forming these metallic structures known as chimneys. And they're spewing out temp waters of temperatures up to 400 degrees from the top. And most of the time, they're still liquid because of the immense pressure from the very deep sea. So first, I would like to talk about these two bivalves, these two giant bivalves that were the first species found at vents. So one of them, the mussel, is Bethmodius thermophilus in the mussel family, Mytilidae, and the other is in the deep sea family Vestromiidae, which is the sister family of what we know very well from shallow water, the Varanids. And these are very large animals. And immediately when you dissect them, what you can see immediately is they have gigantic gills. So the gill actually takes up most of the body, and everything else seems to be mostly very reduced. And this is also the case with these giant clams, where if you open them, so first of all, note the bloody scene, and that is because the clams actually have hemoglobin instead of hemocyanin, like most mollusks. And this hemoglobin is used for very effective oxygen transport around the body. So again, the gill is huge. And the reason why they have such a big gill, later they soon found out, is because they have they have symbiosis. So these are gills are organs where they keep live bacteria inside their cells. These bacteria, instead of using light for photosynthesis, they can use the oxidation of some chemicals like hydrogen sulfide, methane, and hydrogen gas, which is an energy producing chemical reaction. And they can use that energy and turn that into sugar, which is what these animals rely on. So they don't actually need to eat. So they just need to sit there in the water that contains hydrogen sulfide and methane, and then the bacteria inside their body actually just makes food for them. So this is all they do. They just relax in the hydrogen vent fluid. And in hydrogen vents, the ecosystem is effectively feeding on Earth because this compound comes from the seawater going down to the Earth magma in fissures on the deep sea floor, gets superheated, and in the process, they become chemically altered to carry these hydrogen sulfide, methane, and also lots of metal. And this water, because it's hot, so it comes up and then it comes spewing right out of the seafloor again. And this energy effectively comes from the magma. So the animals that rely on chemosynthesis from this energy source are feeding on Earth instead of the sun. But vents are characterized by low species richness because animals need special adaptation to live in these extreme habitats because hydrogen sulfide is usually very toxic to animals at at this kind of at this kind of at this kind of environment. And these species therefore cannot live anywhere else because other places in the deep sea don't have this uh, energy source that can feed them. So the anatomy of gills, you can see the gills are mainly formed of these bacteriocytes, which contain lots of bacteria in both species. But there is a difference in that the mussels still have cells with microvilli, which means they can still filter feed to a certain extent, where the clams can't. So there's actually difference in energy acquirement levels, and the mussels, if the vents die, they can survive for some time from filter feeding, but the clams can only survive if there is active venting at the seafloor. So this is similar to how corals for symbiosis with algae to get energy and nutrition, but instead of light energy, they use chemical energy. And in the case of clams, they actually get the hydrogen sulfide from deep sea fissures. So they get the hydrogen sulfide from the foot and actually takes into their blood and transport it to the gill where the symbiont lives. Whereas the mussels just get them from the gill. So this is why the clams need this super adapted hemoglobin to more effectively transport chemicals. And these are among the fastest growing deep sea animals. So the growth rate of the vent mussel and the vent clam, so these two, are actually matching that of the shallow water mussel, Mytilus californitus. Whereas normally deep sea clams, because of the energy restriction, they grow very, very slowly. And the clams have another very special adaptation, which is that they have maternal or vertical transmission of symbionts. So if you look at their eggs, so this darkly stained area is actually signals stained by bacteria. So the mother packages part of her bacteria to each egg, and the egg can carry the symbiont with them when the larvae travels across the seafloor until they settle. So they actually have the symbiont ready to go from the moment they hatch from the egg. And the mussels are different, and the mussels don't have this. So they have to get the symbionts locally where they settle as adults. So the two species seem to be doing similar things, but actually there are some quite different adaptations that allow them to be good at what they do, how they want. So this means the clams are able to live where there's deeper source of hydrogen sulfide because the foot can stick in deeper, whereas the mussels can't. And the clams can effectively ensure that the babies actually have the ideal symbionts that they have evolved, co-evolved to live with, whereas the mussels have to rely on the environment having the kind of symbiont they want. So next, let's talk about what other species live get for dinner. So we now know about 600 hydrothermal vents worldwide, but there's also what we call cold seeps or hydrocarbon seeps. So these are also where chemically enriched water come out from the deep sea floor, but they are not hot. So these are cool fluids that are about the same temperature as surrounding seawater, maybe 2 to 4 degrees in normal cases. And they also host a similar suite of animals, but often the species that live as seeps don't live at vents and vice versa. And they also are found in globally, and there's actually seeps at both active and passive plate margins, which means they are
Mm.
around everywhere in the world with more than 500 active sites that's known to date. And another type of chemosynthetic habitat is whale falls, where dead whale carcasses, after they sink to the floor, because of the rich oil contained in their bones, as they decompose, they release hydrogen sulfide. So this is something that's not from the earth, but actually this is a source of food that comes from the surface, yet it provides the same kind of chemicals required for chemosynthesis. And therefore, a similar suit of animals actually live in whale falls. And this is similar case with wood falls. It's just instead of whales, you have wood. So they're both big enough biomass to sustain these animals. Anyway, so in all cases, sites from different regions have different animals. So those from the East Pacific, where we just looked at, they have giant tube worms and giant clams. But in Indian Ocean, for example, there's more shrimps and there's iron snails, which we'll come back later. So this scene looks completely different from the East Pacific, and this is the scene from the Indian Ocean. And actually, about a third of all macrofaunal species at chemosynthetic ecosystems are mollusks, or actually just a little over a third. And of the 8 extant classes of mollusks, 7, except cephalopods, are known to have vent or seep endemic species. However, most are gastropods and bivalves. And totally, there is about 400 species endemic to vents and seep added together, with about 200 of them in each. And some groups are quite diverse. For example, the gastropod superfamily Abyssocrasoidea is almost all endemic to vents and seeps, and they are quite diverse at 50 species, and not all of them are symbiotic. So the two species in the middle,
Mm.
Alviniconcha and Ephemeria, they are large snails that can grow up to 10 centimeters, and these two are symbiotic in the same way that the mussels are. So they have very similar way of hosting the symbionts in the gill as the mussels do. And their gill is, again, very, very big for their size. But there's also weird things like Rubispira, which is an endemic of whale falls. So it is only found around dead whale carcasses where it seems to be ingesting whale bones as the only source of nutrition. And then there are more normal species, such as Provana, that live around these mussels and clams, and they see what they do is they graze off the bacteria and detritus from the rots and the mussels, and they survive just by eating detritus and bacterial matter. So if you look at a phylogenetic tree, so you have Provana, which are grazers and detritus, and then you have them later moving on to symbiotic species and also the whalefall Rubispira. So we think Provana represents probably a more pleomorphic type. So what it eats is probably more like the ancestral state of their ancestors. So we use X-rays to look inside the body of how they're adapted to their food. And this is very similar to how we get X-rayed when we need to go for inspections. But what we do is we use synchrotron X-ray, which is a super high energy version of that, by using particle colliders that can provide super bright X-ray light. And the same as cameras, the brighter the light, usually the better the image quality. So we use these big facilities to give us high quality CT scans. And the specimens are mounted in fluid, so the specimens don't dry up, and the scans take about 30 minutes per specimen. And this is the kind of data we get. And we get thousands of slices across the animal. So without having to make manual sections, we can see the internal structure. And by manually reconstructing the internal anatomy, we can see what the inside of these animals look like. So this is Provana, and this is Rubispira, and this is Ephemeria. So you can immediately notice they're actually very different. But what you notice is that their configuration of the anatomy, actually, so how many loops the intestine does and where it goes to, is actually all very similar. So the position of the gill is generally the same, the position of the heart is generally the same. But there are major differences in proportions. So if you think Provana as the plasmomorphic form, and you look at Rubispira, you see the stomach and the hindgut, the intestine is super expanded and contains lots of bone fragments. Whereas in Ephemeria, the gut is actually very, very reduced. And instead, what is enlarged is actually the gill. The gill is very, very big. And this is very obvious if you map the proportions. So green is digestive system, and the blue is the gill. So in Provana, there's this kind of normal proportion. And then in Rubispira, the digestive system massively expands, whereas it reduces greatly in Ephemeria. And on the opposite side, the gill is not that different between Provana and Rubispira, but the gill is gigantic in Ephemeria. So their organs proportions reflect adaptations to their different food source while maintaining an overall similar kind of formula. And this is a very important adaptation that they have in different animal groups to adapt to hydrogen vents. So they devote energy and resource only to what is important to them. So if you graze bacteria, then you have a typical gut and gill. But if you have endosymbiosis, you need to have a big gill where the symbionts are, so you can more effectively get more energy from more bacteria. So you enlarge your gill, but you don't have to eat, so you reduce your gut. And if you eat whale bones, then your gut has to be enlarged because you need to crush and dissolve this bone, which is a very difficult substance to eat. Whereas your gill is normal because you don't have symbionts in them. And next, I would like to talk about the scalyfoot snail, the species covered in iron. So this is a magnet with a specimen of the scalyfoot snail, and it's probably the only mollusk that you can move magnetically.
No.
Yeah, so this is super cool.
Mm.
So it is a literally metal iron snail. And it also has anatomical adaptations where it has a super enlarged heart and a large gill, but there's no symbiont in the gill because it does something quite different. So it has a super gigantic heart and gill, but so this heart is one of the largest heart in animal kingdom. So we actually wrote a paper called Heart of the Dragon because I think it has horns, it has a giant heart, and it's not scales. It's like dragons. Anyway, but what it does is actually it has symbionts, but in a different part of the body. So it has symbionts in part of the gut instead of the gill. But what we are mostly interested is, of course, the scale, right? The iron. Like, why is it covered in iron scales? Of course, it's for defense, right? Everybody think it's for defense, or they thought it was for defense. And then suddenly there was another population that was found, but this population if you look carefully, it's white. It doesn't have iron. So the two are the same species. So genetically, they are identical, but actually, they don't always have iron. And a hint to this is that the vent fluid where the black one lives is 58 times more saturated in iron compared to where the white one lives. But if we do elemental mapping of the scales, we see that there's no iron in the white one, there's only noise, whereas there is iron in the black one. But both of them contain considerable amounts of sulfur. And the iron that's on both the shell and the scale are actually iron sulfide. So it's not just iron that's in the scale, so it's iron and sulfur. But in the case of the white one, the iron is gone, so it's only sulfur. And also, if you think it's for defense, what is it defending it from? Because in some localities where the scalyfoot snail is a dominant part of the community, there is no predator for it. Like nothing eats it, so why does it need to protect itself? Is it really protection? So we looked at the epithelium that's making the scale and what is actually secreting. And if we do elemental mapping, it's very clear. So this is the inside is the foot tissue and outside is the scale. So which means this layer is the newest scale that has just been secreted by the animal. And it's very rich in sulfur. So the entire new scale part is very rich in sulfur, but there is no sulfur inside the tissue, the soft tissue anymore. So the sulfur is actually being pushed out. But if you look at iron, actually the newly produced scales, even in the black population, does not have iron. And iron is actually more rich inside the tissue than in the scale that was secreted. So actually the scale material is originally rich in sulfur, but it's not rich in iron. So the iron must have come later, after the animal secreted this scale. So to look at the specimen, we use a technique called focused ion beam SEM, where essentially you have this tiny micrometer scale specimen of the scale, which then you slice through using this beam of irons. So we use a gallium beam to slice through. So each slice you take a picture, and each slice you take a picture. So in the end, you get an image across this block of specimen, just like CT scans. And so we have a distribution of this is the iron sulfide, and so this is inside of the scale, and this is towards the outside. So you can see there's a gradient of iron forming. If the iron was secreted by the animal, it should be homogeneous. But if there's more iron on the outside, that hints that the iron is actually coming from outside. So what we think is happening is that the iron is actually coming from the hydrothermal vent fluid that's diffusing in to the scale, and it's reacting with the already secreted scales, sulfur inside the scale, to form iron sulfide. So the for the animal, the iron is actually just a byproduct, and it's not important for it. And what's really key for the animal turns out to be the sulfur. And we did an experiment to check whether this is true by getting scales from the white population, and we left it for 2 weeks in where the black one lives. And then the same scales turned black within 2 weeks. And indeed, they did produce the same kind of nanoparticles inside the scales. So the iron is not secreted by the animal. And what we think it does is because it has symbiotic bacteria that uses hydrogen sulfide, and hydrogen sulfide, after being used, turns into sulfur compounds, which is toxic. And the animal needs to do something to get rid of the sulfur. So some symbiotic bacteria actually has vacuoles that they can store these extra sulfur that they can use for energy later. But the scalyfoot female does not do this. So the host, the animal, has to do something. And they basically, after using the hydrogen sulfide, turning it into a sulfur compound, we think they deposit this into the scales as a detox mechanism. So actually, the scales has a complete different function than what people thought it would be. So this is one of the weirdest adaptations for vent animals and totally unexpected. So this is why it's very cool to study these animals. And next, let's just briefly come back to Ephemeria, the same episocracoidean snail that we talked about, because it also has these warts on the foot. So these warts were actually discussed as warts in the original description, but nobody studied it since for 3 decades. So is this part of the foot's soft tissue or is it hard tissue? Because the having scales was sort of like a unique feature of the scaly foot snail. So why does this thing also have scales? And so looking at the anatomical structure, it is quite different from the scaly foot because each of these scales are not uniform, but they're formed by several pieces of the scale stitched together. And also, if you look at the sections, it's a super weird scale for a chitinous structure because chitinous structure, like the scale of the stelyfoot that we have here, so usually there is one layer of secretory epithelium, and then it's directly secreted by this cell. So it's a uniform layers, and they're directly outside epithelium. But for the ephemeria, so the secretory epithelium is very far away from the actual scales, and there are a few layered structure, and then the nucleus that we can see here seems to be gone as you move upwards or outside towards the outside, which is something that was never seen before for this kind of uh, chitinous structure. And what is this very similar to is the picture on the right. So on the left, you have the scales of the Ephemeria snail, and on the right, this is something that's very similar, but from a complete different animal. So this is actually human skin. So human skin makes our keratinous layer on the outside through a process called cornification or keratinization, where cells that are rich in keratin would basically die and get compacted in, into hard structures as they go outwards. And there's this several stages of change in the cellular structure. So first they lose the nucleus, and then they become flattened, and then they become compacted. And this is basically the same kind of stages we see in ephemeria. So this is so these are each cells, or we think are cells, and that are getting compacted to become the outer scale layer. And we checked what material this was formed using X-ray techniques, also at synchrotrons. And then basically, based on the peat profiles, we can identify them as chitin. So they're not keratin, they're chitin. But this kind of structure was never seen before in any chitinous structures. So it's a new way for making chitinous structures, and it's actually analogous how we make our skin. So this is a super weird thing that it does, but we still don't know what this does. It must be some kind of adaptation because it costs a lot to make this scale. But one hint is if you look at the external surface of the scales, there's a lot of bacteria that seems to call this home. And so the function is probably again some kind of symbiosis. And this was a similar kind of thing was actually described from a solenogaster, a worm mollusk found at hydrogen vents. So this is the worm mollusk Heterodomania, and on it has cuticles on the outside, as all aplax do, and then they also have these bacteria inside the cuticles. And today we still don't know what it does, except we can only guess that they're for symbiosis. So they still do something weird that we don't understand. And next, we have a snail that metamorphoses in a very weird way. So this is the snail Gigantopelta in the same family as the scalyfoot snail, but it lives in different place. This is in Antarctica, so this is Gigantopelta chessoia from Antarctica vents. And It also has symbionts, but it's actually not mostly closely related to scalyfoot. So it seems to have evolved this ability of having endosymbiosis independently from the scalyfoot snail twice within the same family, which is also unusual. But what's most weird about this animal is if you look at the anatomy of this animal through growth stage. So this is a 2.4 millimeter baby. So I want you to concentrate on this yellowish green part, which is the endosymbiotic bacteria hosting organ. So first, it's very small, and it's still very small at 3.5 millimeters, still kind of small at 5.1, and then boom, it becomes huge at 7.8 millimeters body length. And then it stays the same until adulthood. So suddenly, at some stage of its life, it decides to switch from normal feeding. As we learned, the proportion of the organs are basically a way of telling what kind of food it eats. And suddenly, there's an expansion between 5 to 8 millimeters body length. So suddenly, it goes from grazing bacteria to hosting endosymbiosis. And when you look at the cross sections of this symbiote hosting organ, at 2.6 millimeters body length, there is no bacteria inside, whereas it's full of bacteria when you look at an adult. So there is a diet shift that occurs after the larvae settles on the seafloor. And after a while, it suddenly decides that it wants to switch the food it eats. And it's very clearly illustrated in the profile of the volume of the body parts. So the solid gray, light gray line is digestive system, excluding the symbiont hosting organ, the trophozoome. And suddenly, this black line, which is where the symbiont is, increases dramatically between 5 to 8 millimeters. And this shift in diet and feeding ecology is switching the niche. And it's not even linked to sexual maturity because the snails we found were actually already sexually mature before the shift. And so this is a kind of new metamorphosis that happens after settlement, which is normal metamorphosis into juveniles from larvae. And it does it again, but it's in a way that's not obvious from the outside. So we call this the cryptometamorphosis. And if your snail grow isometrically, it will be like left. But in Jaganpelta, the organs grow very differently from normal snails. So it's probably easier to relate that if you have a small lizard that eat normal food growing up to 3 meters, and then suddenly it decides to use nuclear energy like the Godzilla and grows to 100 meters. The next weird mollusk is we're going to talk about Dragon Ball. So bear with me for a second. So this, what we're looking at is an aplacophorans mollusk in the class Caudofoveata, and is actually the first Caudofoveata that was found in chemosynthetic ecosystems. This was only described last year from a cold seep in South China Sea. And it was only found by us pushing this 50 centimeter long core into the sediment. And this sediment is very black, so it's totally anoxic. And you can see here, so these are the caudophoviates sticking out at the bottom. So it lives very deep inside the sediment. And turns out this is the second largest caudophovia species in the world. And this was selected as one of the worm's 2024 top 10 marine species, partly because I named it after the wish-granting dragon in the manga Dragon Ball, which is popular in Japan. It's got lots of scales. It's like has a dragon type of body. So anyway, not too dissimilar. But the real surprise was when we dissected one, because we couldn't find a gut. there was only one coiled organ surrounded by spongy material. And if we look at the 3D anatomy of this thing, so this is the external muscles, and we look inside its body. So first we have the gill, which is like normal photofoveates, and then pericardium. And then this winding thing we saw was actually the gonad, and the gonad was covered in the spongy digestive gland. But there is no intestine, there's no stomach. There's only the digestive gland, all the way from mouth. And if we take a section here and look what this is like, so we see there's a lot of bacterial signal. So this is a bacterial probe that was designed to fluorescent when bacteria is present. And if we look closely, it's not everywhere. The bacterial signals are coming from these specific spheres or balls inside the tissue. And we've never seen anything like this before. So these tiny spherical structures, that's where the bacterial signal is from. And outside of these balls, there is no bacteria. And this is very weird because normally the cells are full of bacteria everywhere. And when we made transmission electron microscope scannings of these balls, we found that they are actually spheres that contain a very densely packed symbiont uh, cells. And the walls are actually made of 20 to 30 layers of membrane, which is super, super unusual. But there's no nucleus inside this thing. And we decided to call this structure Dracosphere, which is literally Latin for Dragon Ball. And on the right, you have a 3D reconstruction of the symbiont cells tightly packed inside this Dracosphere, which we think is a new organelle. And why does it have this? So we have a theory, is that because the oxygen level is super low inside the burrow where this thing lives, it's the animal, of course, needs oxygen at all times to breathe. But the symbiont is actually capable of breathing without oxygen because it's anoxic, but it's capable of anoxic respiration. And but if there is oxygen, it will use it, preferably.
Bloody
So
hell.
what the animal wants is the animal wants to use to keep all the oxygen for itself. And it doesn't want the symbiont to take the oxygen from it. So it's making this super tightly bound layers. So the more membrane, the harder for oxygen to diffuse. So they make this 20, 30 layered membrane so that the oxygen doesn't go into the bacteria, but is instead preferably taken up by the mitochondria of the host cells. So the animal can still breathe when they are very much deep down in the anoxic mud and make sure the symbiont can use the anoxic respiration pathway instead. So this is a theory. It's not entirely proven, but this is what we think the best guess that we have for what it does. So it evolved an entirely new organelle to adapt to this anoxic environment, which is not unheard of, but still very, very strange. Some loriciferans, we know they actually turn the mitochondria into a different organ called hydrolysosome when they're in very anoxic environment, and they can actually live without oxygen. But this is a totally different kind of evolutionary pathway that allows the animals to live in anoxic environments. And in the end, I just want to talk about these animals are currently needing of protection. So I'll tell you a story when rewinding back to 2016. We had a research cruise in Indian Ocean where our aim was to study actually how the iron is formed in the staleyfoot snail. And at the time, the staleyfoot snail was known from three populations, and we wanted to go to all these sites. But And then we realized we couldn't go to this one because this place was under a mining contract by the government of China. And then later, this Tyree field was also assigned to a mining exploration license under Germany by the International Seabed Authority. So the only place that the Scalyfoot snail was safe was the solitaire field, which was inside the exclusive economic zone of Mauritius. So it wasn't in international waters, and therefore it couldn't be given a mining exploration license by the ISA, which is a part of the United Nations. And we see this news about deep sea mining in newspapers, in the news program, in radio, and we think, oh, it's probably sometimes in the future. But actually, it's now because the deep sea mining testing has already started in some parts of the world. And what they target is the chimneys. So the chimneys, as spectacular as they are, they're also, if you take a piece of it and look inside, it's super bling bling. And when it's bling bling, it's good, right? So it contains a lot of useful minerals, mostly like iron and zinc and copper, but also gold, silver, and rare minerals. So this is something that will be useful for the mining industry. And essentially, we are already mining hydrothermal vent deposits, but only the fossil ones, because the fossil hydrothermal vents are already an important source of these metals on land. So in Japan, they're known as pure coal, which means black ore, and also they're important mines in Ontario, in Canada, and a significant source of world's copper, zinc, lead, gold, and silver. And then if there's mineral ores, then there will be people who want to mine them. So the International Seabed Authority has granted many exploratory licenses to contractors in international waters. And in the Indian Ocean, we have China, and we have Korea, and we have Germany, and also India, who's interested in all of these sites. And the mining code, which sets the regulations for eventual mining activity, has been delayed for several rounds, but it's due this year. And some countries are also interested in mining their own EEZ. So in Japan, the first successful test mining for hydrothermal vent minerals was actually the first in the world that was done in 2019. So this is an actual scene from 2019 when the Japanese test mines bulldozed the seafloor for hydrogen vent ores and then extracted them to the seafloor, from the seafloor to a ship like this. And for example, earlier in Norway, in January 2024, Norway approved deep sea mining, including hydrogen vent deposits, which was suspended later, but we don't know when the suspension will be to. And unfortunately, Indian Ocean hydrogen vents are actually very little known. So we actually have very little knowledge about what happens if mining happens. There is significant scientific knowledge gaps for evidence management. And cold seeps are no exception because cold seeps are typically associated with gas hydrates. So if you see these cold seeps, they're usually bubbling, and this bubble is some gas hydrate, most likely methane hydrates. And sometimes you can directly see the methane hydrate here, the white specks, right next to the animal colony. And gas hydrates are essentially, you can use them as replacement for natural gas. So they're being eyed for mining as an energy source in the place of natural gas. And in Japan, methane hydrates already been test mined since 2013, and it's thought to be more commercially viable than even hydrogen vent sulfide ores. So coming back to Scalyfoot, so this is when we realize that they're actually threatened because if mining takes out two of three of its populations, then it's very, very threatened. But how do we tell the world about this? And it's not just the scalyfoot that's threatened with extinction. More than 40,000 species are. And we know that through the IUCN Red List. when we talk about endangered species, everybody know what endangered species is. And they know this because the IUCN Red List assessed them as endangered. So you know a tiger is endangered because of this system and because of IUCN. And so how do we get a species to be assessed under the IUCN Red List to say that it's really in danger? And IUCN functions through specialist groups, which are all volunteer experts that manage their own different specialties. And there's over 10,000 experts around the world. And so they're mostly actually taxonomic group specialists. So we have coral specialist groups, we have dragonfly, we have wild bee, hornbill. Is hornbill enough group to have a specialist group? I don't know, but anyway, there's a hornbill group. And then there's the IUCN mollusk specialist group. So it seems to be doable to assess mollusks. And there are 5 criteria: population size reduction, geographic range, small population size in decline, and very small or restricted population, or through a quantitative analysis showing a decline. And importantly, being on the red list doesn't mean a species is threatened because actually half of the assessed species are least concern, which means they're not endangered at all. But it's important to assess them in the criteria that's defined in the IUCN Red List. And so, can we apply the IUCN Red List criteria to the scaly-foot snail? And after looking at the criteria, we think because in the deep sea, we actually don't have enough data to discuss population size reduction because we don't go to these sites often enough. And so therefore, we don't actually know how much decline it is in right now. And we don't have enough data to do quantitative analysis. But what we do know is the geographic range of the species and that the population is very small because vents are small. Hydrothermal vent fields can be 1,000 kilometers away from their nearest neighbor. So they are like deep sea islands, and these animals cannot live anywhere else. And each vent field is only a size of the football field or even smaller. So we looked at this specific criteria, and which allows us to actually assess a species based on a plausible future threat. And if the number of locations where a species is known from and the geographic range is small. So if we apply this to the scalyfoot, we know each vent field is less than a football field. So the total range of the scalyfoot in 3 vent fields is 0.02 kilometers squared. And area of all known potential Indian Ocean vents, including those we haven't discovered, but only known from plume signals from the ship, add up to less than 1 kilometer squared, which is actually a lot less than the 10 kilometer square defined. And we know the scalyfoot from, at the time, from 3 locations, which is less than 5. And obviously, the threat status is corresponded with mining potential, because if a place is likely to be mined, then that is what is threatening the species. So if you put an area of occurrence inside a marine protected area, then there is no mining potential and therefore the species is not threatened. Simple enough. So if you look at the area occupancy, so 2,000 kilometers square is vulnerable, 500 kilometers square is endangered, and 10 kilometers square is critically endangered, and the scalyfoot is 0.02 kilometers squared. But the minimum allowed in the IUCN Red List assessment is actually 4 kilometers squared because they require by 2 kilometer buffer. So, and also another question is how do you define a location of occurrence? Because when you mine a hydrothermal vent field, you create this plume from bulldozing the seafloor. And how far this sediment plume can travel is actually important in defining how much impact this mining event will have. And in the literature, we see that the mining activity could spread across 60 to 70 kilometers. So we suggested to use a safety buffer of 80 kilometers as one location. Because if you kill one vent in the middle, the other two might also be covered in these cuttings from the drilling and then therefore die. So if we look at this matrix that we came up with, the scalyfoot lives in less than 5 locations and lives in a place with mining license. So therefore, it can be from endangered to vulnerable. And in the end, the scalyfoot snail was assessed as endangered in 2018. And this was, of course, the first species assessed as endangered due to risk of deep sea mining. And this was published in Nature Ecology and Evolution and reported by 100 plus news outlets, including BBC, Guardian, and also Nature and Science. So it was useful. And also the scalyfoot snail is widely known as icon of the deep sea, especially in Japan, as a popular species with many merchandise being made, and it helps raise awareness. So these are some of the toys that they make in Japan of the scalyfoot snail. And we actually produce lots of pins and other items for the scalyfoot merchandise in Jamstack. And the scalyfoot has also been featured in popular Japanese comics that sold over 22 million copies. And also we had collaborations with Nintendo to feature the scalyfoot. So this is the scalyfoot girl in the game Splatoon, which has sold 10 million copies around the world. And also Animal Crossing, which was super popular game in the times of COVID when people couldn't go outside. And we actually had a collaboration with them to include scalyfoot as one of the decorations you can have in the house. And also, you might have heard of Pokemon, which is a super big thing. So in 2022, if you remember, I have alluded the scalyfoot to a dragon. Nintendo actually made uh, a Pokemon partly from the inspired by the scalyfoot. Its metallic shell developed as part of the mucus of the skin reacting with the iron in the water, which is exactly what the scalyfoot does. And this, and interestingly, this species has a steel dragon type, which I think is perfect for scalyfoot. And we make lots of merchandise also for expeditions at the Natural History Museum in Japan. So the scalyfoot is great as an umbrella species for raising awareness of these deep sea animals who are nearly extinct. But we didn't stop there. So Calyptogena magnifica is now nearly threatened because it's known from 9 locations, but one of the locations is in marine protected area, but the 8 of them are not. So it is not least concern and is not endangered, but it's somewhere in the middle. And Gigantic pelta is least concern, and the other species of Gigantic pelta is critically endangered. Why the difference? Because one of them, the Antarctic Gigantic pelta chessoia, lives actually within an NPA and Antarctic Treaty. So one of the sites is actually south of 60 degrees south. It's covered by Antarctic Treaty. So there is no way that this species would get mined. So therefore, it is least concern. Whereas the other species is only known from one location with a mining license. So that's why it's critically endangered. And we completed assessments for all vent mollusks, which included 40 critically endangered and 33 endangered and 43 vulnerable species. But there's more to do because, for example, Chidoderma shenlong, like we don't know if this seep species is endangered because we haven't assessed it, and or other species like the giant tube worm. And therefore, there is a new Venn red list project aiming to red list all named habitable Venn species, and not just mollusks, because we were limited by the mollusk specialist group only assessing mollusks. But now, through the Sentient Bird Ocean Species Alliance, there's a launch of a new marine invertebrate red list authority that allows us to assess other invertebrates as well. And we are aiming to assess all described Venn endemic animals, so nearly 500 species within a few years' time. So there has been some events around conferences around the world, and we have different experts for different taxonomic groups to help us. And currently, we're in the process of proposing a new specialist group that expands this effort to code seeps. So we're proposing the code seeps and deep sea hydrothermal vents IUCN Red List group. So watch this space. And at that note, thank you very much for inviting me. And I would like to thank all my collaborators. And I'll take any questions if you have some. Thank you very much.
Thank you, Chong. That was absolutely marvelous. What a load of different things you've covered there, and in such detail as well, with so many exciting innovations. Kind of mind-boggling, really. You said you were going to blow our minds, and I, you can count me in.
Thanks.
Uh, yeah, just amazing. Um, does anybody have any questions? You could raise your hand using the React thing, or just physically do it, but we might not see.
Can I ask a question if nobody else does?
Yes, of course.
These organisms occur in these very isolated deep sea vents. How do they get from one vent to another? I mean, how did the separate populations arise?
Yeah, so like the shallow water mollusks, they mostly have dispersal in larval stages. So a lot of them disperse by plantotrophic or lesotrophic larvae that travel. So if you're on a ridge, then there is a current along the ridge that the larvae can be transported across. Or if you're in ocean basins, there are also currents that can move you around the basin or rarely outside of the basin. So some of these larvae can live up to years. So this allows connectivity to other vent sites. And typically, the different vent sites in a region where this species occur form metapopulations. So vents sometimes depends on how fast, depends on the technological activity level of the area. Sometimes the vents are only living for a few decades and then a vent will die, but a new vent will arise. And then the larvae from other places will settle in this new vent, and then all this network of vents form a metapopulation that sustains the genetic diversity of the animal.
Yeah. Thank you.
No problem.
Um, I have a question if we don't have any others at the moment. Um, we've got one on the chat from Victoria Burton. Um, how much do the communities between the events overlap? And are there likely to be many more species to discover?
So different vents in different regions have are in different vent biogeographic regions. So usually within a small area. So for example, parts of the Indian Ocean. So we now have three provinces for the Indian Ocean. So at that kind of scale, many species are shared, but species rarely cross these boundaries of provinces because of dispersal barriers. So, for example, if you live on a ridge, then if a deep transformation fault exists that breaks the current moving larvae across from one ridge and it doesn't go to the other part because there's a break in transformation fault, so the highway stops there. And that kind of thing will stop larvae dispersal, and so effectively making the species divergent across time. So then they become different species over time. And yes, actually, there's lots of species being discovered every year because we still haven't been to many of the hydrogen vents around the world. So, for example, Indian Ocean is actually very poorly known compared to the East Pacific, for example, because deep sea exploration is expensive and there's no rich countries around the Indian Ocean that has been spending a lot of time and effort on exploring Indian Ocean ridges. Until recently, actually, it's quite ironic, but it's because of this mining licenses, more countries have been spending money to explore Indian Ocean vents, and therefore we are just starting to get a better idea of the diversity in Indian Ocean vents, for example. I hope that answers the question.
Thank you. Yeah. Another one. Oh, yeah. Thank you from Victoria. Another one from the chat. Uh, what would you say is the most challenging thing about studying deep sea species, other than the location?
Uh, Keeping them alive. So bringing up deep sea animals from thousands of meters deep, usually so at least if you're a fish, then you burst, right? But at least for mollusks, they don't have a gas space inside the body, so they are actually alive when you take them up, but they have cellular level stress. That means they're not healthy. And for most species, it's really difficult to keep them alive for more than a week or two. And so doing experiments on them is quite difficult. And that's, I think, the hardest thing about studying red animals.
Hmm. Um, I've got one quick question. It's just I was wondering how old these vents and vent systems were, and there are fossilized vents billions of years old in Japan that are being used for mining. Um, so I guess there are no fossils of animals associated with any of those extinct systems, which is why they weren't discovered until the 1970s. Is that right?
Yeah, so we didn't know any fossil animals from vents before that, partly because actually we didn't know vents existed. So, for example, like even if you just talked about extant animals, so some species of the Vescomyid clams, some Trypogenus species were actually described before vents were found, but they're only from dead shell fragments, and nobody knew where they were from, and nobody's ever seen live one. And in terms of fossils from vents, there's, I think, three sites around the world that fossil vent shells have been found. But the difficulty is because hydrogen vent fluids are also acidic. So any shells and materials will rapidly dissolve in hydrogen vents, which means they have a very slim chance to get fossilized. And so, even in these three sites, the shells are casts of sulfide minerals that surrounded the shell, and the shell is completely gone. You just have the casts, and not very many either. So, actually, most of the fossils that we have for vet and seep animals are from seeps, where the fluids are less acidic. So, a lot of seeps actually fossilize quite well. So, this is partly how we know most of the groups that we see now are either arose around Jurassic or Cretaceous.
Right. So they're kind of like they're like living fossils, but there are no fossils, I guess. That,
Yeah, but they're not really living fossils because actually because there was a huge anoxic event around that time, which killed off most of the vent animals that were there. And so the animal groups we see now are mostly actually newly evolved since the Jurassic. And
okay.
so, for example, instead of bivalves, they used to have lots of big brachiopods at vents, but we don't have any of them now. And it's all completely taken over by bivalves.
Wow. That's brilliant. Thanks.
Can I ask another one?
Yes, of course.
I mean, the evolution of these things must be quite extraordinary. They're completely different mechanisms of life than some things we know of nearer the surface. I mean, is it possible to study the evolution at all? I mean, we don't have any intermediate species or anything.
Yeah, so one thing we can use is so sometimes there are like for mussels, we have other close related mussels. So this is one specific subfamily, but we also have comparisons from other mussels. But this is actually quite rare. So in many cases, we can only assume that the species which have a more normal lifestyle, like grazing or detritus feeding, are more plesiomorphic, and use that as a model to study how the more extremely weird ones came about. Does that answer the question?
Yeah, I think so. I mean, basically, we don't know enough yet.
Yeah, it's quite challenging, but we have to make some assumptions here and
Yeah,
there, unfortunately.
yeah. Thank you.
Thanks.
Okay, thank you very much. If there are no other questions, I would like to suggest we all thank Chong in the usual way with a round of applause, which you can unmute yourselves for, of course. And
Yeah, thank you very much for inviting me.
no, thank you very much, and thanks for providing a primer on IUCN threat categories as well, which uh, will be useful for our society coming up, I think. Um, that was marvelous. Thank you very much.
Thank you.
Thank you very much.